salute. So this week, it's my pleasure to introduce our speaker, Arturo Sanchez-Azoviva. Arturo is a STRI research associate for what, 20 years or now? A long time, anyway.
12 years.
12 years. He's been working in the cranes in Panama at least 20 years. But anyway, Arturo is also a professor at the University of Alberta. He's originally from Costa Rica, and he has a long history of research in tropical dry forests and also some wet forests, especially ecophysiological research and remote sensing research. And today he's going to be speaking about 20 years of research on lianas and trees at the STRI cranes. What have we learned? So, Arturo, take it away.
Yeah, and one note before we start for people who've just tuned in, we're going to save questions to the end unless there's something like really urgent. And to ask a question, you can raise your hand and then I'll monitor and call on people when the time comes.
Okay, let me see if I can start this thing. I have my talk here, sorry. I hate this technology.
No worries. We have we're still, the number of people joining is are still joining, so the delay is okay.
Oh, okay, yeah, because I cannot see my, when I try to do the Zoom share in the I cannot see my PowerPoint presentation.
You should, so under share content, it should say screen as one option. So you might just try share screen.
Yeah. Yeah.
Or, oh, sorry, no, I'm looking at it. Sorry, I'm looking at an iPad. It probably looks different to you.
Let's see. Hold on, let me start this again. I'm gonna turn off the quick PowerPoint. Okay, hold on. I'm gonna close everything that I have here and go from there. Okay, so let me see, because we did the test this morning and it was working fine. So, okay, here we are. Okay, let me see, go to Zoom. Okay, here you go. Perfect. Okay, perfect. Do we have it there?
Yes. Yeah, we
Fantastic.
see your PowerPoint window.
Okay, well, let me just first start by thanking Ellen for the opportunity to come and give a talk about the work that we have been doing in Panama. And as a matter of a history of how I got engaged in Panama, it was very funny because it happened 20 years ago in the ATPC meeting in Bangalore. And Kamal Bawa asked me, Arturo, you need to talk to this guy, George Wright, that is from Panama. And I said, oh, okay. So we met in India and we ended up working in Panama. So the first trip that we did to Panama and the cranes starting in the year 2000, and it has been almost 20 years of collaboration. So we are very happy of the work that we have been doing there. And I just wanted to share with you some of the work that we have been doing over the years with Joe and many of my past and current graduate students. So let me see. Okay. So the first thing that when I started working in Panama, I was getting into the cranes and looking down, of course, and then I saw this thing. I got fascinated by trying to understand how could we detect lianas from space. That was a question that has haunted me for many years, many doctoral dissertations and so forth. And the view from the crane provide so many insights about tropical forests. When we look at the publications of many authors over the years, lianas in remote sensing tend to be ignored. I really don't know if because they're seeing more like an ecological ground level work, like some of the great work that Stefan Snitzer has been doing over the years, and Joe and others. or the fact that the people working in remote sensing seem to be extremely focused just on tree level or a landscape level, and tend to ignore the presence of lianas in the landscape. So we start working on trying to answer some basic questions. And in 2004, a paper by Lewis and al. Show up and start talking about the significance of lianas that got my attention. We know that they are parasites, and they are very well adapted to disturbance. And their theories are really, some people say that it's because age effects, lianas are increasing. Other people may think that it's a result of disturbance, mortality, and so forth, that lianas are increasing. There are also some theories about the relationship between CO2 concentration increases and lianas that are more prompt to grow faster on the higher CO2s. We have, in fact, a paper that we actually, Joe and I, many years ago, and I should not say this, but I remember picking Joe at the airport in Edmonton, and he had in his pocket a bunch of liana seeds, and that we grew here in Alberta on the controlled CO2 chambers. And absolutely, definitely, you can see the difference in CO2 concentrations, but also very picky. They don't like to grow they didn't like the temperature, they didn't like the photoperiod here in Alberta, so it got very difficult to grow them. But those that grew under double CO2 really, really grew faster and with a higher density of leaves. So there are many different theories about why they're increasing, but what we know is that they are systematically increasing across the tropics, and including in BCI and Costa Rica and in Brazil. Simon and others also in 2004 identified 10 potential drivers of environmental change in the tropics. And one of those was the increased liana numbers, and among increases in precipitation, climatic extremes, etc. So that's probably why we have a very interesting question to answer in terms of what are we seeing from the ground, what are we seeing from the near surface remote sensing, and what we are seeing from satellites in terms of liana increases. This is a very interesting paper by Smithsonian from 2005 that actually helped us to set up a little bit of the work that we have been doing. Stefan presented this about 14 years ago, more or less. And I remember reviewing with him some of the initial drafts of the paper. And we can see that we have an inverse relationship between precipitation and liana abundance. So the higher the precipitation, the less lianas and the more trees. And so that makes it very interesting to work in the Panama Isthmus, because we have Parque Natural Metropolitano in the Atlantic, sorry, in the Pacific, that is drier. And we have Fort Sherman in the Atlantic that is wetter. And we can see it when we look at mean annual precipitation for both sites. In Parque Natural Metropolitano, drier site, and in Fort Sherman, we have less lianas and also more trees. So we have been working for, I think since 1998, on Mexico, Costa Rica, Brazil, etc., on liana issues. And I'm going to focus this time on the work that we have been doing at the cranes over the last 20 years. And as I mentioned before, Parque Natural Metropolitano is drier than Fort Sherman, so it's a very interesting contrast in terms of what we can see. We have more lianas in the Pacific, less lianas in Fort Sherman, and vice versa in terms of the trees. So this is more or less what I was thinking, the sequence of work that we have done there. This is more or less what we have been doing. And at the time, some of them were really crazy, I got to admit. I'm very interested in the physics of light. And I told myself, I cannot understand how lianas can be detected at the canopy level, unless we have an understanding of first the physiological and cellular behavior of light when they go through a leaf. So that's our first initial work, started in the year 2000. Then we start moving slowly as equipment became more available in terms of hyperspectral properties of lianas. Since the year 2000, also we have been exploring machine learning and other very advanced classification techniques to try first to separate trees and lianas at the leaf level. And because we didn't have the technology, we couldn't find a plane, we didn't have drones, and the spectrometers were too big. Then in 2009, with Microsoft and others, we started working on the use of wireless sensor networks, and with Joe, we deployed a bunch of different wireless sensor nodes in the canopy of Parque Natural Metropolitano to look at the temperature changes and differential changes between liana leaves and tree leaves. So that's what we started working on this. And then that evolved with as soon as we have better cameras in the use of thermography and thermal mapping. And then right now, as we having a better spectrometers that we can mount in drones, and lighter systems, we're actually looking at the analysis of ecosystem structures, the liana infestation at the cranes, which is a process that was stopped by two things. One, the crane that need to be fixed, and the second, the pandemic. So we have been pretty much stopped at the thermography component, but they're gonna put some ideas forward, some of the work that we would like to do. Okay, so let's first start by thinking about how a liana leaf looks compared with a tree leaf at the microscopic level. And for those who are not very familiar with the cross section of a leaf, we have been actually getting cross sections. It has been insane work. I remember going up at the crane with Myrna very late during the day, putting the leaves in a cooler, getting to the first plane to Alberta, and it was like a marathon. In the moment that someone got into the airport, hand the cooler to another person and running immediately into the lab to do cross section. So between collection and the first cross sections, we was about less than 24 hours sometimes. So it was a lot of work, but we are very interested in looking at leaf thickness, mesophyll layer, because in fact that's in this section in the mesophyll layer is where you have a light scattering, and it's controlled by the near infrared. And then we had the vertical palisade cells too as well, who played a very important role that we will see later on. So this is some of the examples of the histology analysis that we have been doing. This is for 2009. We start getting 36 liana species and 20 tree species. And we collect the sun leaves. And those more or less are different examples of how the liana cross sections look like at this. One of my favorite ones is this one is Diolocarpus majors, and then Anacardium excelsum. This is very, very fantastic trees and lianas to work with. So the first thing that we noticed was that systematically across both grains, the liana leaves were thinner. This is data collected during the wet season, so there are no water stress. They're fully grown, and we can see that in the Parque Natural Metropolitano, lianas are thinner than the trees, and the same thing in Fort Sherman. So that was a very interesting thing, and it started actually posing some interesting questions about how they behave, like in terms of how much reflect light. Then we continue with one of my past graduate students, Dominica Harrison, and to analyzing with Joe a little bit more the thin sections that we have. And we start finding, confirmation. Lianas have in the Parque Natural Metropolitano and Fort Sherman, they have more empty spaces inside. So basically they tend to release more temperature, so that's why they eventually may look hotter in terms of emissivity. And also when you look at the density in the palisade cells and in the mesophyll cells, then definitely lianas again are different than their cousins on the trees. And both present a statistical significant difference, both in the number of, in terms of the density of those cells. So basically explains a little bit more how lianas may actually reflect in light differently than, in a different intensity than trees' leaves. So when we have this kind of information, we start asking ourselves, okay, how this reflects as a leaf overall? Because light comes, hits the surface of the leaf, goes through it, interacts with the chlorophyll, interacts with the empty spaces, and either goes through or comes back into the atmosphere. So this is an example for those who are not familiar with the spectral reflectance of vegetation tissue and what controls the factors. So we had the first section between this web length, and this is reflectance between 0 and 1. And then we have this is what we call the visible. This is what our eyes can see between 400 and 700 nanometers. So we have blue, green, and red. And this is the green peak, or where we can actually, 550 nanometers, what I think that the leaves look green. This is basically controlled by chlorophyll A, chlorophyll B, sensified cycles, and so pigments and so forth, carotenoids. And then this area right here, this significant increase, is actually related to the two things: water content, leaf age, and the amount of empty spaces that are inside of the leaf where the light goes through and actually reflects inside of the cells and actually contributes to having a higher or lower reflectance. When you have a leaf that has very little empty spaces, this thing goes down. And when you have a leaf that has a lot, this thing goes up. That's pretty much the control that we see in a very simple way. So when you look at the pigmentation in the visible, the first thing that we noticed was that, again, it's very interesting. Lianas in both cranes tend to have less chlorophyll concentration than tree leaves. So we can actually see this in the dry forest, but we didn't saw that in the rainforest. It's something very interesting to see that there is this differential in terms between the Pacific and the Atlantic cranes. Now, which is very interesting is this is consistent. If you have less chlorophyll, you have more reflectance at 550 nanometers, and we can see it right here. So the lianas in the dried area have a more reflectance at 550 nanometers, and the trees have less reflectance. So we can see this in the dry season and in the wet season, so it's very consistent. But in Fort Sherman, we don't see absolutely any of this. They're very stable, extremely stable. So those differences in terms of chlorophyll concentration are statistically significant. So that was one of our second findings in terms of how lianas reflect light. Not only that they're thinner, but also that some spectral indices were more dominant in the dry forest than in the wet forest. We were later on able to verify this in Mexico, in Costa Rica, and in Brazil. So it's very systematic. But if you look into the rainforest, you don't see any difference in chlorophyll concentration. So this is an example again, and I'm sorry that this came a little bit broken, but you can see these curves right here. This is a Parque Natural Metropolitano, the average tree spectrum. And in the top, we have the Parque Natural Metropolitano average Liana spectrum. So they tend to reflect more in the visible. This is very interesting. This is for a paper that we published in 2004. And we kept that thinking about this long time ago. And we applied this observation to Costa Rica. at the Santa Rosa National Park, and in a paper that we published last year, where we use deep learning classifiers, and that we actually train our hyperspectral data to this kind of knowledge. And actually, we were able to get a very high accuracy in the detection of the extent of Liana coverage when we actually tell them that the Lianas we have pockets. When you have a very pocket of a lot of reflectance of light of 550 nanometers, maybe that you have a pocket of Lianas. So this is what we trained the classifier to look at, learn from the process, and then we went back into the field, and we got about 85 to 90% accuracy in terms of our predictions. So it's a good indicator that these processes or these observations from Panama can actually are consistent and can be applied to other sections in the dry forest. In terms of spectral indices, which actually some of them are controlled again by chlorophyll, we can see that some of them are true. For example, lianas in the dry season and in the wet season behave different than trees, and they have more variance and so forth. So basically, this is very typical for single ratio, modify single ratio 705, and modify NDVI 705, very consistent. This is the liana during the wet season, for example, in the ND 705, and this is the tree wet. So basically, we can see that lianas in the dry forest are very, very consistent in terms of those differentiations spectrally. Meanwhile, in Fort Sherman, we don't see that difference at all. I don't know if it's because and I think that probably more people like Joe and Stefan did said, I don't know if there is an element that there are less hydric stress, or there is a more adaptation to heat or so forth, but systematically we are observing this across many different spectral observations that we have. When we try to classify this, and for example, trying to relate chlorophyll concentration versus the different indices, we can actually see that they separate extremely well. And this is probably we were very successful in using machine learning approaches in the dry forest. Meanwhile, in the wet forest, we cannot find absolutely any differences. And that came out in 2009, where we suggested the possibility that we may be having a Liana syndrome that is typical of dry forest ecosystems. And the idea was that we could try to test this in other sites. I mean, we had been very consistent. We got to Santa Rosa, we got to Chamela, we got to Mata Seca in Brazil, and all the lianas followed exactly the same pattern. And when you go to the Amazon, everything is lost. So there may be that this syndrome is real, and it's going to be probably more work coming along just to see if we can actually characterize better this kind of behavior. Again, if, for example, if we use elements of classification in machine learning, like using the first derivative of neural networks of K nearest neighbor, we can see how well the mean trees separate from the liana in the spectral space, the principal components. So we can actually separate them very well. Meanwhile, when we look at the rainforest, nothing. It's just a mixture. So what are we learning from here? I think that from my point of view, what we are learning is that if we want to have a monitoring system to identify the impact of climate change on liana extension, it may be more successful in dry forest environments than in wet forest environments. That's to me something that is coming very clear, and that's what we have been trying to pull together over the last 20 years, is how we can use remote sensing tools in dry forest to actually detect the fingerprint that these colleagues from England were mentioning in 2004. We may not be successful in rainforests unless we expend a significant amount of time and effort, and I don't think that we have just the technology to look into that. But for dry forests, maybe something that we can actually use as a tool to validate those effects. So by 2009, we learned a lot. 10 years ago, we knew that lianas have significant differences in chlorophyll concentration, carotenoid concentration, water content, leaf thickness, and also that reflected also in specific leaf area and in specific spectral indices. We didn't notice much difference in the fourth Sherman, but that's why we can see that the only thing that is consistent is that leaves are thinner, and the specific area, of course, will be different between trees and lianas in both sites. But for the rest, the key dominant element is, again, dry forest. So probably that's why we think that doing the exploration in the dry forest could be better. We can notice here that the reflectance at 800 nanometers there is no difference, and confirms Harrison work when we see that the percentage of the empty spaces in lianas and trees is not different, and that is also a reflectance here in at 880 nanometers, which is no difference whatsoever. So that's what we learned about by 2010, 2009, and then new tools starting to emerge, and Joe and I started exploring wireless sensor networks, and we went to the crane and we put a few sensors on the thermocouples under the leaves. And I remember going to the crane, sitting in the center of the crane, launching the stuff and downloading all the data. And this is what we are observing for the experiment. At that time, we were learning a lot how to install it. The technology was really early in its development, so we have a lot of problems with water condensation and so forth. Today, we don't have that problem because we have learned a lot in the last 10 years. But what is very interesting is that from the original data that we got, this is a presentation that we have in the IEEE eScience conference in Stockholm in 2011, that we're observing that lianas were a little bit warmer. The leaves of the lianas were a little bit warmer than trees. So we parked that because we couldn't do a lot of work on this because the technology was not available. And then a new camera system show up by FLIR, and we bought one, and we started working on 4 species: anacardium, trees that were infested with lianas, anacardium, delwea, the Castilla elastica, and anona at the crane in Parque Natural Metropolitano. And what we did was around noontime, we went into the crane with the camera, aimed the camera at 1.8 meters from of optical distance, and we took pictures, thermal pictures, and the camera has the capacity to take red, green, and blue or regular photos, and on top of that has the capacity to take a thermal photo in Celsius. So we concentrate, or we did this mosaic, that, by the way, this mosaic is online, and it has a 5mm resolution. You guys can find it in Gigapan. And then we can actually go and look at the trees and actually take thermal photos, like the one that you guys can see from Castilla elastica. So we were able to get the red, green, and blue, or the visible, and then the thermal one. And then we're able to go and query. Now, one of the problems that we have when we start working with thermography, when you look at the use of these cameras, is that everybody goes with a 0.98 emissivity. And talking to one of my graduate students, Jose Antonio Guzman at TICO, that is finishing his PhD with me, we were asking ourselves, lianas are thinner, they have more empty spaces, they have more water. What do we know about the emissivity of lianas? And I was talking the same thing with Joe. And we designed this experiment to estimate the epsilon of lianas in a series of samples that we got of species that were running into the crane. And basically what it is you take like a bucket and you put at the bottom aluminum foil with like a very thin layer of water. And then you put your leaf on top, and then you take a thermal photo. And then with the reflectance observer, we can actually estimate the emissivity of a leaf. And this was crazy. Trying to do this we were actually doing it in the bathroom of a hotel for many, many hours. And we came out with this, and it's very interesting because many people use the emissivity of 0.98 for vegetation, they forget. But we can see that there are some trees, the trees like Anacardium, Annona, and Castilla elastica have less the emissivity, not exactly 0.98, 0.975. Then the Luea is a little bit closer to 0.98, it's probably the only one. And then the Lianas have a great variability, going all the way from 0.98 to 0.99. So there are statistical difference between the different ones. So this was a very interesting finding in terms of how Liana emit light. And we actually were able to go back then to our photos and calibrate the photos for the real emissivity that we were observing, which in fact affects the temperature. And since we did that during the wet and the dry season during 2015, 2016, we were able to see that the difference between at the leaf level and at the canopy level. And at the leaf level, we can see that the anacardium stays relatively stable. The Annona changes. We couldn't find the Annona this year. I think that the tree failed. Joe will confirm with me, but I think that that tree that we were looking at fell or died. And then in terms of Castilla elastica, we can see that there are some changes in terms of Liana tree leaves, but the statistical difference was not. So what we're finding out, and the conclusion of this was, yes, Liana's leaves are warmer, but they are not statistically significant at the tree leaves. They're a little bit warmer, but not as much. And the same thing, we'll see this when we compare this with the crowns, although her hydric stress affect that reflectance, that emissivity. One of the things that we would like to do, well, we have not been able to do it because the crane was being repaired, is to use a new type of spectrometer. Because what the thermal camera does is takes just a thermal picture and give you temperature, but it doesn't give you the reflectance that in the 8 to 15 microns, which is where what we call the thermal infrared. We have done those experiments in Costa Rica, and we published a paper last year on remote sensing of the environment that demonstrates that you have enough spectral structure in the 8 to 15 nanometers to differentiate between lianas and trees. But we have not been able to do it in Panama yet. So as soon as we have a possibility to travel and the crane is operational, we would like to bring that spectrometer to actually test these observations that we have in Costa Rica in Panama, and actually start looking closer at what kind of compounds control that reflectance in the 8 to the 15. For you guys to know, we go from 0.4 to 2.5 microns. That's what the commercial spectrometers are looking at. And what we're looking is far, far away in the thermal, 8 to 15. So there is a potential thing that can emerge there, but we're still waiting to learn about this. So all of this is just going to the crane in the wet season or in the dry season and measuring something. So at the time, there was, in the world of remote sensing, came the concept of spectronomics. And everybody, or a few people were saying, not everybody, I sound like Trump, but a few people were saying that if we actually get a signature of a leaf that goes into a spectral library, that signature is unique. And that signature can be used to train classifiers. And for example, you have the spectral signature of Annona or Enterolobium cyclocarpum, Enterolobium, or any other tree. You can actually go back into an area where you flew your instrument, and then you can actually detect those trees. And the question came back from Kamal Bawa, can you really detect that tree or not? So the first thing that we actually, because we learned a lot about this seasonal effect, went back and we look at very carefully at the spectral properties of the leaves as a function of season. And we try to use, I'm a hydrologist by training, and we use a agglomerated classification algorithm that basically what it does is you put a bunch of different spectrum and then it gives you this kind of decision trees. And we have the same thing in hydrology when you look at the concept of network complexity. And we can actually use a spectra for different species. And we actually can run in MATLAB this agglomerated classification algorithm that actually produce graphs like this one that show us where or how the different species accommodate themselves when you run this. And we published this in 2012. And what we did was we used the spectrogram of the lianas. This is how, if you put the lianas, looking from the top to the lianas and trees for Sherman, and lianas and trees for Parque Natural Metropolitano. This is how the combination of a spectrum look in the wet season and in the dry season, and the difference at the species level. So you can see that there are some that show difference and some that they don't show differences from mostly look like a DNA graph, but that doesn't tell us enough about how this information can be. It's very nice, it's what I call a wow factor, but the information that you obtain from this is not very useful. So when we use this kind of classification algorithm, and we start looking at all the species, we say, wow, look at this. If you take Fort Sherman during the dry season and run your spectral data, and then you look at all the species in Fort Sherman during the wet season, the networks that you are developing are totally different. If the concept of spectronomics will apply, like has been presented, those two dendrograms will be the same, and they are not. And we can see this also for Parque Natural Metropolitano during the dry season and the wet season. The dendrograms are statistically different. The locations or even some species in the dendrogram vary at the end. And we can see this is for Fort Sherman, and this is for Parque Natural Metropolitano. You guys can see, for example, this is in the dry season, this is in the wet season. There are significant difference here. And even the position is also different. And when you look at lianas, it's also the same thing. So they tend to classify different. So the question is, if I go and collect data today, can I use that data that I collected today, spectroscopy data, to classify data collected 6 months ago? And we have seen papers of people that collect data today and then get another data set 4 years later and try to combine things. And this is what we're observing. And this is a very interesting graph. This is the classification accuracy using machine learning approaches for lianas and trees and all the species together. So if you use data from the same season to classify your data, let's see that you go into the crane, you collect your data, and then you fly a drone with the hyperspectral data. So you may have the chance to detect, to have very high accuracy of samples. But if you go, let's say that you collect data in the wet season for your drone and use data from your dry season, your accuracy goes absolutely out the window. The same thing can be seen right here. For Fort Sherman in the dry season, very good. But if you use data from the wet season to classify dry season, then it goes off the window. And the same thing. Basically, you need to have data from the same, or literally instant, or concurring with the data, with your remote sensing data acquisition, if you want to make sense of any potential monitoring approach to look into the lianas and trees. And basically, to me, that also brings the concept, or the question is, can spectral signatures and spectral libraries for specific vegetation work? In my opinion, they don't. You have to collect your spectral libraries at time, or your drone collection, or your remote sensing collection. Otherwise, you can get into this kind of problems and very high levels of error in your classification. And basically, the last part that I wanted to talk is about near surface remote sensing. And this is just for all the colleagues that are very interested in drone work. And this is the latest work that we did at the Parque Natural Metropolitano before we had the crane going down. But what we're trying to do right now is to actually see how these crowns, for example, these crowns that are infested with lianas, and one that they're not infested with lianas, reflect light. And so this is an example. I think that we collected about, if I'm mistaken, Jose Antonio was telling me more than 1,000 spectral signatures for those trees during the dry season and the wet season. This is an aggregated value. And we can see, of course, the first thing that we can see here is the effect of water on the classification, and or on the data. We can see that it goes down, so it's less water, so this element comes down. And if you actually look closely, canopy with lianas tend to have higher spectral reflectance in the visible than in the near infrared. Also, we are observing this peak right here. This is a water absorption feature that can be extremely interesting. And the question came back here and say, well, what happens if we do measurements every meter? We put the crane and we put the spectrometer at 1 meter, 2 meters, 5 meters, all the way to 10 meters. So with Elwin, we were actually measuring the impacts of the field of view. So what we have here is 5 meters and 10 meters between the floor of the crane and the top of the crown. And we can see that the height, the effects of height is not significant yet, but we need to process the data. This was acquired 2 years ago. And about what is very interesting is that when you start looking at the spectra at the canopy level, at the NDVI, we can start seeing really interesting separability using full spectroscopy data. So this is very exciting to me, because if you look at the new sensors that are coming out, like Evan Broadband from the University of Florida, that can have this capacity of combining LiDAR and hyperspectral, flying the drone very close to the canopy could be very interesting to actually detect those species, or not those species, but the extent of liana cover. So we have trying to use this technology combined with deep learning classifiers. And what we're trying to do right now is training these algorithms to look at many different observations, looking at what we learn about the spectral indices, what do we learn about the NDVI. And we actually bring these into a hidden correlation model in the deep learning classifier. And then we can actually start getting the true extent of the liana coverage. And this is very exciting. And I was telling Joe, wow, we can get this done every year. And we can compare every year maps, and we can see what the two things, the extent and the rate of extent. So this is what happened when the crane went down, because it couldn't be repaired, and we have to get a new one. So that's what we stopped. And it has been a long way to try to get that crane operational, but hopefully it's going to be ready. So we have learned so far by mid-2020, literally 20 years after we start this journey. We can see that some leaf level reflectant properties are being transferred to the canopy level, and they're independent of the height measurement. This is very encouraging, because it can help us to identify better classification approaches as we get more and more embedded into machine learning techniques. And these new classifiers that either can be neural networks or it can be deep learning or machine learning or whatever you want to call it, can be automated as we saw before. And it can actually help us to detect the extent and the rate of change of extents of lianas. And I cannot believe, for example, there are still people doing the stuff by hand. And we need to move away from moving this. There have been actually papers that suggested that we should be continue measuring liana extent by drawing this in the canopy. And I think that this is not what this is about. We need to have consistent and reproducible data sets and approaches that can help us to do long-term changes. I also want to say that I think that the future, what we are observing in Costa Rica right now, is not in the visible and near infrared. I know that this is going to be tough for many people to think about it, but we need to start thinking beyond those wavelengths of 400 to 2500 nanometers. We need to look at the 8 to 14 microns. There is a sensor from the US Navy that has been flown already, and with this kind of thermal properties. And the spectral structure that we are seeing right now, and combined with machine learning and other algorithms, there is very powerful. I think that that's probably the future of species identification in the tropics. We will probably see something very, very powerful coming in the next few years. And of course, we need the cranes back. So finally, my last slide is on emerging opportunities. Where do we see this coming? I think that there's great excitement in the integration of LiDAR and multi-hyperspectral remote sensing. I think that the work that Evan at Florida is fantastic, and I think that he has been doing some work in BCI. Then the evaluation of drone-based thermography, putting spectral cameras, they're thermal, not that the ones that measure temperature, because this is a mistake. You have to get a camera and measure radiance. So you can actually do the transformation, the mathematical transformation from radiance to temperature, which is probably the best way to approach this. And also the integration of terrestrial laser scanning with airborne drones, LiDAR-based systems is also very important, because as the experience are telling us, the TLS doesn't go, it misses about, in BCI, I think that we are missing about 5 meters in the crowns, and in Parque Natural Metropolitano about the same thing from the bottom to the top. So we can actually complement from the top to the bottom with the LiDAR, combine both of them and have a better cloud points. And also the role of lianas on essential climatic variables, photosynthetic radiation, temperature, relative humidity, soil moisture, using advanced soil in sensor networks. I think that Joe has some of those, and Helen in BCI, and we have about 40 of this running in Costa Rica right now. So we will see what happens over the years. Well, okay, this is pretty much what I wanted to show you. A lot of people have come to work on this over the years, and it would be unfair not to thank Joe because he is the force behind the support for this, all the way from permits to getting us booked to use the crane. So thank you very much, appreciate it.
Thank you very much, Arturo. Sorry, it's raining here now, so there's a little bit of background noise. So if people have questions, remember you can go raise your hand. Oh yes, we have a question from Ori Sanhur. So go ahead, Ori. Ori, you should be able to. Yep. Okay, we can hear
everyone. Arturo, thanks so much for the great talk. Very interesting. I have a couple of questions. First, with regard to your first part of the talk, when you talked about the difference between patterns in dry versus wet forest. Is that something that you found regardless of species? I mean, this is something that it's happening. all Liana species?
No, not all Liana species. Some Liana species behave very similar as STRI species. The drier the environment where you have the higher number of lianas, the more difference that you see. Some of the species are very, very different in Parque Natural Metropolitano, but some are not. The biggest difference that we have been observing are in Brazil, in a site called Parque Estadual da Mata Seca in Minas Gerais, when you have 900 millimeters of average rainfall per year. So, and also in Chamela too as well in Mexico. So the drier, the more difference between lianas and trees, and but no, there is no, we cannot come back and say all lianas. When you pull them together, yes, but when you look at the species, not all of them are different than the trees, and that makes that even complicates a lot of the things, because imagine at the beginning of my talk, you have this picture of a liana, and you have, for example, an Acardium excelsum leaf pulling out, and you have the liana around. And sometimes, when I was talking to Joe, there is one tree in Parque Natural Metropolitano that has 27 species of lianas. 27 species in one single crown. So the level of spectral mixing that you have, that's so massive that it makes the problem so complex. It's not linear. It's just a level of complexity that is so difficult that we try to get everybody rather than individuals.
Thank you. The other question I have, if I may, is the last part of your talk, which I agree is very exciting, about that ability to be able to quantify sort of the liana extension over the canopy. And you talk about machine learning. I was just wondering sort of the kind of equipment, the capacity that is needed to be able to do sort of the technology behind our ability to do what you just expressed, considering that we know that you're studying that and you want the cranes back, but once we have the crane back, so what technology is required to be able to do this kind of research?
Well, in terms of technologies, we have the spectrometers, and I think that Joe still has a box in his house or something like that, and there is one spectrometer there. We have like 7 or 8 spectrometers that we constantly take to Panama. So in terms of the spectroscopy equipment, there is no major issues. I mean, we can share it, but at the end of the day, what we need is fast computers. And I think that the average price of a machine that you need to run this can be around $10,000. And it has to be very fast, humongous amount of RAM, 2 terabytes of hard drive at least to start. And I'm gonna give you an example. We did the terrestrial laser scanning for BCI, some sections of BCI for Joe, and also for Parque Natural Metropolitano. And the data from PNM is so massive, but so massive. It's billions of points that we require to have an infrastructure. We have to go and buy a new computer that is absolutely dedicated to that data set alone to be able to be run. So another thing that we have been very successful, and I think that is probably good for you guys to know, is the possibility to access to supercomputing systems. Some of the data that we run, I think that one of the first papers that we wrote with Joe and Karen Castro in 2004, I don't know, Joe, if you remember, we used the Canadian supercomputer system in BC, in British Columbia, to run the data just on leaf data. So the you need fast computers, high capacity storage, and RAM. If that's what my students are constantly asking for. 256 gigabytes of RAM going up, the faster the better. And also, probably they can correct me, but they're constantly asking me for R and Python and all these different things that they can actually run. But yes, you need to have a significant infrastructure, 2 levels of infrastructure, I would say. One, to access to supercomputing systems. We use the Munich one, the supercomputer in Munich, and the one in Canada, the supercomputer system. And but we have also dedicated computers with very high graphics machines to be able to run some of the data sets, because some of the stuff that we have, the PNM data is so massive. The TLS data on PNM is so much that we're running any laptop. It's just super crazy.
So we have a couple other questions, people asking questions. Stephanie, you want to go next?
Sure. Great, thanks, and good to see you, Arturo.
Nice to see you, Stephanie.
And enjoyed your,
years.
yeah, it has. Great to hear a synthesis of what you've done at the cranes, and really exciting to see you've developed, starting to develop algorithms that can automatically detect trees versus lianas. And I think using the deep learning approaches obviously have really good sides and sides that are harder to interpret. And so one question I have is, do you have a sense of what it is about lianas versus trees in the Parque Metropolitano images that allowed them to be separated? It's probably not leaf level properties, but something about canopy structure. And I'll just end by saying, I think one exciting thing about the deep learning is that it has a lot of potential for cross-site effectiveness, whereas some of the other techniques don't. So I'm also excited when the cranes get back up to see if these algorithms can work across different forest types as well. So main question was about what factors about the lianas versus trees do you think is driving your ability to separate them?
This is a very good question. We are going to throw something very crazy here. I think that it's a combination of both, Stephanie. Surface structure, the matting of the canopy, and also the properties of some specific spectral properties of some of those leaves. So it goes back, if you have very flat, you have very smooth changes in the variance of the surface combined with a higher reflectance in the near, in the visible 550 nanometers, there may be a signal that you can train in the deep learning classifier to look for that. We are going to start a really comprehensive liana removal experiment in Costa Rica this year, where we have pre and post surface a lighter system both in the ground and from the top. And we are trying to characterize that structure, the complexity of this surface structure, pre and post harvesting or pre and post cutting, to try to start seeing if we can see if in fact that stuff that you are mentioning, and what I think also I agree with you, could affect the separability of lianas. And one of the things that we would love to do is to have a drone flight by Evan and his drone in PNM, but it's always very difficult because of the proximity to the airport and the crane. But I think that you're correct. But to me, it's a combination of things, and we cannot separate them yet unless we have a very important control experiment. We were trying to do the same thing in the Amazon with the liana removal experiment, but with this pandemic running there, I don't think that we're going to get even permission to fly to Brazil over the next 2 years here. the university is actually prohibiting all international travel right now for the next year. And I could probably travel to Panama, probably travel to Costa Rica, but I don't think that will happen in traveling to Brazil. So that probably will have to put to park for a while. I agree with you, Stephanie. I think that you're making a very good point. That is that a structural element plays a very important role. Although I really haven't looked at yet at good data to be able to actually say yes or not, or it's 80% or it's 50%. It's a combination of things.
Okay, we have another question from Bill Weislow. Bill?
Yeah, hi. I guess my question was kind of along those same lines, if you can hear me okay. And it was what I was going to ask was rephrasing it a little bit, and I was wanting to know if you knew anything or could speculate perhaps about either the physical properties of the leaves or the physiological properties of the leaves that might be associated with some differences in such that between whatever it was you said, 8 and 14 microns, that particular range becomes important. And is that spectra correlated with anything that physiologically might be important to the plants?
No, I don't think so. Physiologically, no, but there are some compounds in that region that we have not explored. There has been a paper by the USGS where they did a compound extraction, but I have not been able to follow this on Bill. The paper by Jose Antonio in remote sensing of the environment looked just at the structure of the spectra and the change of the spectra. And actually, it was very interesting, Bill, because when you look at the reflectance of leaves at 400 to 2500 nanometers, everybody looks the same. They look like this, you know, like the figure that I show. Mm-hmm. The only difference between one species and the other is that move up or move down, or if the leaf is new, it moves up or move down. But all of them follow the same spectra. So the game is here is how it moves up and who moves down, and how this relates to physiological elements of carotenoids, of chlorophyll or water content and so forth. But in the 8 to 2500, this is a totally different ballgame. It is an absolute ballgame. The spectra is not the same.
Huh. And then if I can ask you one more that you can feel free to say nah. But here's something that I wonder as we move increasingly towards a reliance on machine learning and artificial intelligence, that as these have been applied to human social behaviors, who gets a bank loan, who gets a job, that it reveals some of the problems with people programming. Yeah. And I wonder how we're going to deal with this in biology, where we have very much less of a preconceived idea of what a biased result would look like.
Well, I'm going to tell you what my problem is with this thing. In machine learning or in deep learning classifiers, you have your input data and your output data. And in the stuff in the middle is a black box. So the neuron connection there is just extremely random. And you don't have control. So how you build those correlations and so forth of these machine learnings or those matrices and stuff like that has very little control in how you do it. And that to me is very problematic because at the end of the day, you don't have control over process. Like if I want to do a supervised classification or something like that, I know that the population has to be normally distributed, but this is like a dog breakfast. You just take your data, you dump it, you say, okay, this is the process that it is, and boom, at the end you get something else. That's the main problem that I have with this process of machine learning. They are starting to become more effective. Some of my students, Ji Xiao and Ji Ming and others have been working on this. And the math is extremely complex on this too as well. So become more interdisciplinary. What we need to actually do is to get this hybrid computer science, biology people that actually, the bioinformatics people that can actually sit down with us and learn more about what we're doing. Not like some of my colleagues tells me, give me all your data and I will give you a product. I say, no,
Yeah.
sorry, that's not
Yeah,
going to
no.
work for me. No, no.
All right, thanks.
You're
We have
welcome,
another
Bill.
question from Oscar Baron Ruiz. Oscar, you want to go ahead?
Hi, doctor. Thank you for your presentation. I just want to ask you some question about the applicability of this research. So, one of all the monitoring you and our team have done, have you seen that the ecological structure of the forest due to Liana infestation has affected the ecological functions of the forest and or decrease or increase the certain ecosystem services?
In terms of ecosystem services, I don't know, Oscar. We haven't looked into that. The work that we did with Branco many years ago showed that Liana-infested forests have higher levels of avian diversity when compared with non-Liana-infested sites. They have more tree diversity, more bird diversity so, but I cannot say water or carbon or arsenic values or anything like that related to ecosystem services. It's a very difficult question to ask. In terms of forest structural components, Stefan Schnitzer have done a lot of work in Panama on the impact of liana removal and the effect that they produce. Also, some of the work that we have done in the lab with terrestrial laser scanning, when we actually have thrown this hypothesis for discussion that lianas tend to change the three-dimensional arrangement of tropical forests. Like, for example, you look at those forests in my colleagues from Europe that, oh yeah, let's use this camera to do these data sets, and then you say, could you please show me where your site is? And all of them are perfect trees, you know, like the tree and nothing around. So this is very deterministic, you know. There's the trunk, the ground, and the tree and the branches cannot be more deterministic like that. But in the moment that you put lianas, that stuff goes all bananas. And what you have is a change in the three-dimensional arrangement of the forest space. So what we're trying to do right now is use the TLS, the terrestrial laser scanning, to scan liana-infested forests, remove the trees, characterize the use of the space by those lianas, and then put the tree backs and remove the lianas. So the question is how this forest will look without lianas. What is the use of that 3D box? And then you put the lianas and you take the trees and you say, okay, how can we characterize these fractals and so forth? So this is something that we would really like to do that. And I was looking forward to this year to go to BCI and do it because they're bigger trees and they're easy to take out. But you know, with this pandemic and stuff, everything went down the window. So it's just imagination and modeling what is left right now.
Yes, thank you. And I have a second question, but related. I want to know your opinion. So, for example, based on the evidence you provided regarding the carbon uptake and aiming at mitigating climate change, do you consider that liana infestation should be directly managed? In other words, like remove some of them from the forest as a tool to increase the capability of the forest for carbon sequestration?
No, I don't think so. I'm a little bit into the don't touch the forest. They are part of the ecosystem. They have been there from day one. The day that you have a tree, the liana came next to the tree. So unless this is a very question for management. If you have a plantation, you want the trees to grow faster. Of course, you need to remove the lianas. And we know that when you remove those lianas, there is an increase in growth. And the review by Stefan Schnitzer in Biotropica is very clear. I mean, of the 99 liana removal experiments in the tropics, 64 were on plantations, and all of them show that the trees grow up when you cut them. But I don't believe that liana removal it should be used as a tool for carbon sequestration control in tropical forests.
Okay, thank you.
So we have a couple more questions. One from Carlos Jaramillo. Let's see. Do we have Carlos on? Let me.
Yes,
Okay.
he can talk now.
Okay. Go ahead, Carlos.
Hi.
Hi.
So imagine you are able to follow the PCI 50 hectare flood for the past 500 years. Do you think that the density of the Lianas over time will be shifting all the time? At the end, every piece of the forest will have a Liana at some point during a lifetime of a tree? So in other words, what is the main predictor of Liana density?
Ooh, ouch. What is the main predictor of Liana density? That's a very important, interesting question. I'm gonna give you my perspective because I work in secondary dry forest. So I can actually answer that question, not for the 50 hectare, but by following Santa Rosa since 1970 when it was created and it was a farm. I believe that there are two things that play a very important role in the liana infesticides that we have in Santa Rosa, and it's past land use. And even Dan Jansen will tell you the same thing. If you have a plot that was a coffee plantation, and you have a plot in the same type of soil that was a rice field, and you abandon them and come back, the level of fertilization that occurred there, for some reason, the one that was rice, you cannot get in. It's so infested. The one that has coffee did not have that much lianas. So past land use plays a very important role, at least what we have learned in Santa Rosa, especially with having the highest density of lianas in intermediate forests. Going back to the question of BCI and the 50 hectare plots, I think that I was gonna ask Helen to answer the question because of Joe, because I never been there, so I don't know, I could not answer that question. I know from the point of view of my experience in Costa Rica and secondary tropical dry forests, that previous land use played a huge role in the density of lianas. But it's only for secondary forests. I don't know, Jaramillo, what can I say about the density? For a long time, I believed that there were trees that liked lianas more than others, like the Guanacaste, Enterolobium cyclocarpum, because I had never seen one single one in Guanacaste with lianas in Costa Rica. And then I go to BCI and I found one. So the whole theory, sorry, I went to Parque Natural Metropolitano, I get into the crane, and the first thing that I see there is an Enterolobium cyclocarpum with lianas. So that's my whole theory going down the drain. So it's very difficult. I mean, why a liana is present in a site? Why they prefer one tree to the other? Why, how they transmit, how they communicate, how they grow. It's just, to me, we focus so much on the trees that we forgot the lianas. And so I think that there's a lot of things to ask. There's a very good question, Carlos. I can only answer from my point of view, my experience in Guanacaste.
Thank you, Arturo.
Okay, we have another question from Eric Manzane. Eric, do you wanna?
Yeah, hello.
Hi.
I have a question. In one of the slides, you show that there is like a less chlorophyll density on the leaf.
Mm-hmm.
So does that mean that there are less assimilation of carbon, less biomass? But there have been a lot of trees showing that lianas are actually outgrowing trees on the dry zones. So how is that relationship happening? It's just because lianas have like deeper roots than trees? Or what is the total effect of this lower density, the lower concentration of chlorophyll on the leaves of the lianas?
I don't think the deep roots are the issue of root depth. I thought about this a long time. I was thinking that the root depth was a very important role. And then you go to Santa Rosa and you find that after 50 centimeters of soil, what you have is a layer of volcanic ash that nothing grows underneath through it. So all the lianas that we have seen in Santa Rosa have grown into the first 50 centimeters of soil and then nothing else. because of the
Interesting.
volcanic ash. That's what the Liberia in Costa Rica is called the White City, because the calcium carbonate that is everywhere. You drive there and you can see these layers. So they don't have deep roots. Trees don't have deep roots. Liana don't have deep roots. This carbon assimilation is extremely important. I agree with you. I think that may be more important in the rainforest than in the dry forest. What causes lianas to have less chlorophyll concentration, be thinner, reflect more light in the thermal infrared, and so forth. The only thing that I have to think about is a liana adaptation. And to me, lianas, I told Joe once that I thought that the apparatus of every liana is like a small air conditioning system. And that when you have a lot of water there, just trying to keep cool, the stomata continue open and close to release more heat into the atmosphere. You have the signal that can be very interesting. It's a combination of things. And so going back to your question, which is a very good one. Now, what kind of mechanistic elements provides for the lianas to have less chlorophyll than the trees? From the point of view of remote sensing, at least to me, was that there was a mechanism for them to be in the top and having to survive high photosynthetic active radiation and higher temperature. But that remains to be explained more by the ecophysiology people.
Okay, thank you.
So thank you very much. So we're 20 minutes past the hour, so I think we should wrap up the questions here. Arturo, thanks again for a very interesting talk, and look forward to more conversations about lianas and trees in the future.
Yeah, and I look forward to be able to go back to Panama and work with you guys again, hopefully very soon.
We look forward to having you here again sometime.
Yeah, as soon as Air Canada can fly to Panama, hopefully we can get the first plane back on there.
And Panama lets Canadians in. Yeah, that's the other part.
Yeah, no problem. And I want the crane.